Hi, my name is Martin Kampmann. Thank you for having me, and I'm excited to share our results with you. I will tell us about our lab's efforts to capture disease-relevant conformations of proteins by deep mutational scanning. And the disease that my lab focuses on a lot are neurodegenerative diseases, which are devastating not only for patients, but also for their families and caretakers, and which are a really urgent need for society because we currently do not have any approved disease-modifying treatments that could really change the progression of the disease. And part of that is, I think, because we still have so many open questions around the mechanism driving these diseases. One thing that has been known for more than 100 years is that specific protein aggregates and specific cell types in the brain are associated with all of these age-associated neurodegenerative diseases. And here you see kind of a rogues gallery of different protein aggregates associated with diseases such as Alzheimer's disease, Parkinson's disease, and others. Of course, one central question that we need to ask is whether these protein aggregates are causal and drive the disease, or whether they're more incidental and some kind of byproduct of sick neurons. And there are two important lines of evidence that suggest that protein aggregation is causal in disease, or something about these aggregating proteins. Number one, we, over the last 10, 20 years, have recognized that for all of these diseases that are usually sporadic, meaning they have no known genetic cause. There are a small number of patients who are who have familial disease and have a disease-associated mutation in the very same proteins that aggregate in the disease. These can be point mutations, or these can be just amplification of the wild-type proteins, or mutations in enzymes that process these proteins. So that from the human genetics tells us that something about these proteins can be driving disease, and in vitro studies suggest that these mutations can make the proteins more aggregation prone. And I think the tightest case for that is Huntington and other polyglutamine diseases, where expansion of a CAG repeat encoding glutamine causes disease, and the length of the repeat correlates both with aggregation propensity and inversely correlates with age of onset of the disease. The other line of evidence comes from studying rare diseases such as mad cow disease in cows, scrapie in sheep, kuru in humans, that are infectious neurodegenerative diseases. And Sten Prusner here at UCSF uncovered that in fact the infectious agents of these diseases is an aggregated protein that can actually in a autocatalytic manner, if you will, template the aggregation of more proteins, and that this aggregated state or prion state, how he called it, is causative in disease. So again, protein aggregation or something about this process can be disease causing. So there are then three important questions that my lab is trying to answer, and many people are trying to answer. First, which exact states of a given protein are disease relevant? Second, what controls the formation of these disease relevant protein states? And third, how is it that these protein states cause disease? And today I would mostly focus on the first question, which protein states are disease relevant? This is a non-trivial question because even though we've made a lot of progress in structurally characterizing, for example, protein aggregates, one feature of many of these proteins associated with neurodegenerative disease is that they can aggregate in different states. Here are some data, for example, showing recombinant alpha-synuclein that can form amyloid fibrils in different conformation or so-called polymorphs. And now through advances in cryo-EM, we even have atomic resolution models of how these polymorphs look. Here's another example of tau, where again, recently there has been a whole set of different cryo-EM structures from tau isolated from patient brains, from patients who have different diseases associated with tau, including Alzheimer's disease, Pick's disease, corticobasal degeneration, or even chronic traumatic encephalopathy, so something that develops, for example, in football players and soldiers after traumatic brain injury. And you can appreciate even from these cartoons that the same tau can actually form aggregates in different states. And so that raises the question, which of these states are, what controls these different states? And if we can have different states, which states are disease relevant? The fact that in a given disease we isolate and can structurally determine the structure of one of these proteins does not in fact mean that that is the structure that causes the disease. In fact, it might be that the disease causing structure is a minority species that would be very hard to find out about using classical structural biology methods. And for a protein like alpha-synuclein, which is intrinsically disordered, in fact, it can take on a number of states. And there have been papers over the years that have argued for a number of these different states being disease causing. For example, alpha-synuclein can adopt a helical conformation in the N-terminal region upon binding to a membrane surface. This can form helical oligomers, but also more disordered oligomers, and of course, these beta-strand-rich amyloids. So taking on very, very distinct structures. And again, the fact that you might find amyloids in a post-mortem human disease brain does not necessarily mean that the amyloid structure is what's driving the disease. You could, in fact, argue that what you find in neurons post-mortem are structures that are found in those neurons that survive the disease, right? So they might even be some sort of protective structure. And even if they're not, it might be that they're the majority structure, but that a minority structure, maybe a more oligomeric structure, might be disease driving. So in order to get at this difficult problem, we deploy deep mutational scanning in the context of a class. This so-called PUBS class, Physical Underpinnings of Biological Systems, was initiated by Jamie Fraser at UCSF. And many of you might have heard talk about the class and the first projects which were into deep mutational scanning of ubiquitin under different environmental conditions and stresses. And when I took over this class from him, I decided that we wanted to leverage deep mutational scanning in order to get at this important questions of which states of a protein that's associated with neurodegeneration might be the relevant species, and whether we could leverage DMS for this. And a huge shout out goes to Robert Newberry, who is a postdoc in Bill DeGrado's lab at UCSF and who served as a course coordinator for this course, really doing a lot of the setup of this whole experimental system, and a lot of the follow-up experiments also together with Bill. And Robert is on the job market right now. So he's a fantastic scientist and teacher as well. And so in this class with students, we of course needed to do something that is feasible in such a context. So we decided to work in yeast. And yeast had been previously used as a model system of alpha-synuclein toxicity, and that had been pioneered by Susan Lindquist's lab. And you may say, well, is yeast an appropriate model system? Yeast does not endogenously express alpha-synuclein, but if you overexpress human alpha-synuclein, it has a toxicity phenotype in yeast. And Susan Lindquist used this for genetic modifier screen, and it turned out that some of the modifiers of alpha-synuclein toxicity in yeast in fact validated in other, including mammalian system of alpha-synuclein. And so we do think that something can be learned from investigating alpha-synuclein in yeast, even though it is not naturally occurring in yeast. So we built a plasmid library of human alpha-synuclein with all possible single point mutations, and introduced this in yeast cells for competitive growth assays, taking samples of the population at different time points and using next generation sequencing to determine the frequency of sequence variants and how that changed over time in order to calculate fitness values. And of course, as you can imagine, wild-type alpha-synuclein is already toxic. So if you had a mutation that makes it even more toxic, the frequency of that mutation would relatively decrease to a wild-type alpha-synuclein. But if you had a mutation that disrupts alpha-synuclein toxicity, that frequency would increase, relatively speaking. And so that's what we were determining in this experiment. And we were quite hopeful that data from this experiment could really help us differentiate between some of these major models that had been put forward for what the toxic species of alpha-synuclein is. Basically, the membrane-bound helix model, for example, presumably requires an amphipathic helix to be bound, where you need polar residues facing the membrane interior and more hydrophilic residues on the outside, potentially even positive charges that stabilize this orientation by interacting with negatively charged phospholipid head groups. And you could imagine that such a structure, if this is the toxic one, would be disrupted by, for example, introducing polar mutations here facing the membrane interior, or reversing positive charges in the plane of the phospholipid head groups, versus the membrane face, the helix face facing away from the membrane would be much less sensitive to substitutions. Vice versa, an amyloid would have obviously quite different predictions. Amyloid surfaces are often formed by nonpolar residues, excluding water at the interface, and very stabilized that way, and obviously have also kind of a periodicity in which residues alternatingly face the different faces of the beta sheet. So we would expect quite a different pattern of disruptive mutations if an amyloid was a toxic structure. And this is then the heat map of fitness values that we observed. On the horizontal axis, you see the position along alpha-synuclein. It's a relatively short protein. And on the y-axis here, you see all the amino acids that a residue is mutated to. We had pretty good coverage, although a few values were missing here in gray. And in shades of blue, you see residues that, mutations that decrease toxicity of alpha-synuclein. And you can see there's a lot of blue. We don't see a ton of red. So it was actually not easy to make alpha-synuclein in yeast more toxic than it already is. As a sanity check, you can see that mutation in the start codon really is less toxic, which makes sense because then we shouldn't get expression of alpha-synuclein. And we also then saw that there was a lot of structure, especially here in the internal half of the protein. Especially, you can see that mutations to prolin along this whole stretch were mostly detoxifying alpha-synuclein. And then mutations with a specific periodicity, mutations to a hydrophilic residue made, or even charge residues, also disrupted toxicity, versus mutations to hydrophobic residues overall had much less impact. And if we then map this to this model of the amphipathic alpha helix, you can see that this starts to make a lot of sense, because based on the periodicity, those residues really that were very disruptive to toxicity were mutations to hydrophilic residues that would map to one side of the helix, and that would be then the membrane-facing side of the helix, versus disruptions on the other face that faces away from the membrane had less of an impact, except to mutations to prolin, which is of course a classical helix breaker. So then our results strongly argued for a amphipathic alpha helix as a really important species for this toxicity in yeast. And plotting the periodicity, you can appreciate that it was actually really the frequency of fitness disrupting mutations, or fitness restoring mutations maybe, was very regular. Here in black is a regular periodicity, and you can see it fits it quite nicely. Now, interestingly, if you ask what is a periodicity, it was 3.67 residues, which actually does not perfectly match the periodicity of an idealized alpha helix, which is 3.6 residues per turn. In fact, if you take this idealized alpha helix and you map this 3.67 periodicity on the surface of that, you will appreciate that these residues kind of migrate around the alpha helix. So if indeed this this relevant conformation of alpha-synuclein was a perfect alpha helix, then the hydrophobic residues would kind of start to drift along the axis and around the axis. And so to put this on a planar membrane, you'd actually have to introduce some strain in the helical turn of the alpha helix. However, of course, one model that would satisfy that is if the helix indeed binds to a curved membrane, in particular, a membrane with a negative Gaussian curvature. And this is very interesting because if you now make this model, this is the kind of type of membrane curvature you might find at a point where a vesicle docks and fuses with a membrane or maybe pinches off from a membrane, which is in fact in neurons the case at the presynaptic membrane where synaptic vesicles fuse with a target membrane. And alpha-synuclein's normal function has been implicated to be at that step, suggesting that maybe this this conformation that we're picking up on here is actually also related to the normal function of alpha-synuclein. So that was very exciting from a yeast deep mutational scanning experiments to get so much quite detailed information about a state of alpha-synuclein that's relevant for toxicity. What it does not tell us is whether this is the toxic state. It's possible that indeed alpha-synuclein toxicity is linked to vesicle binding, and recently, even in the brains of patients in the so-called Lewy bodies and Parkinson's patients that include aggregated alpha-synuclein, vesicles have been detected. So it's possible that this plays a role in humans, but it's also possible that this critical state in some sense is a transition state that enables formation of other toxic species. So you could think of a chemical reaction with a transition high energy state, and that really we're picking up with the deep mutational scanning those mutations that are relevant to be able to populate that state to then transition to the final state that is actually toxic. And of course, the other big question is whether toxicity in a yeast model is relevant for humans. And I think it's really important to think about protein structure in the context of relevant cell types. One feature of neurodegenerative diseases, for example, is that the so-called selective vulnerability. So even people, for example, who are born with a point mutation in tau, and where this mutant tau is expressed in all the neurons in their brain, tau will only really strongly aggregate in specific cell types, or at least that's where it will aggregate first. So really the behavior of a protein and the structure ultimately of a protein depends not only on its amino acid sequence, but also on the cellular context, so interaction with the cellular environment. And so that's obviously then something we need to study in the relevant cell types. And so for that, our lab is leveraging human iPSC-derived neurons, where we can work with patient iPSC lines and differentiate them into the relevant cell types to study the behavior of proteins. Avi Samuelson, a postdoc in the lab, has worked with neurons that we receive from Ligand's lab, or iPSCs, I should say, that have a point mutation in tau associated with frontotemporal dementia versus isogenic wild-type controls. And you can see that the mutant tau forms a phosphorform about 6 weeks into differentiation of the neurons. And this phosphorform is recognized by an antibody that also detects diseased tau in human brains. And maybe even more excitingly for our question, conformation-specific antibody for tau oligomers. This here shown the T22 antibody, but we have now reproduced this with a number of other conformation-specific antibodies as well, detects tau oligomers in mutant but not wild-type neurons already 2 weeks into differentiation. And importantly, Avi is able to couple this to flow cytometry, and thereby you can see the mutant neurons are shifted to higher levels of tau oligomers. And so on that basis, we can now conduct, for example, deep mutational scanning in a high throughput screen based on flow cytometry sorting. The deep mutational scanning part of this is still ongoing, but I'd like to share with you what we're already doing to work on the other side of the equation. So not only looking at amino acid sequence determinants of aggregation, but the cellular environment components of aggregation. And for that, we established a platform to do CRISPR-I and CRISPR-A modifier screen in iPSC-derived neurons, where we can use, for example, CRISPR-I to effectively knock down expression of proteins in neurons. Raylin Tian, a student in the lab who now started his own lab in Shenzhen, developed this in collaboration with Michael Ward's lab. And we can do very high throughput screens for neuronal survival, for example, introducing guide RNAs that target different genes for knockdown. We can have complex readouts such as single-cell RNA sequencing, and we've also conducted some arrayed screens. So Avi can now use the system to target, for example, all protein coding genes in these iPSCs with a tau mutation, differentiate them to neurons, and do FACS sorting to separate neurons that have higher levels of this tau oligomers or lower levels of tau oligomers as stained by this T22 antibody, and then use next generation sequencing to quantify guide RNA frequencies in both populations. And so the same scheme will apply once we do this by deep mutational scanning. And so just to show you that this is really working, on this volcano plot, we have 20,000 genes and their phenotypes. On the right, increase in tau oligomers. On the left, decrease in tau oligomers, knocking down MAPT. Tau itself was one of the top hits where we get fewer tau oligomers, so that makes sense. It's a kind of a sanity check. And other things that make sense is that knocking down autophagy factors, for example, builds up tau oligomers, and autophagy is thought to be a clearance mechanism for protein aggregates, versus knocking down mTOR, a negative regulator of autophagy, is beneficial and reduces tau oligomers. And in fact, small molecule screens have found also that mTOR inhibitors that induce autophagy reduce tau aggregates in neuronal models. Looking in an unbiased way, what types of hits we get a lot of hits associated with stress response, and then a lot of genes in the mitochondria, electron transport chain, and protein synthesis, and almost all of them increase tau aggregation when knocked down, and we can phenocopy this with compounds that inhibit the same genes. And so that's an interesting area, because neurons are thought in the aging brain to experience loss of mitochondrial function and reactive oxygen species, which leads to tau misfolding. So now we might have a cell-based model for that. And of course, molecular chaperones were the other big class of proteins that we thought might come up, and we found a few. And we can now validate using biochemistry that knockdown of these chaperones increases levels of high molecular weight tau and insoluble tau. And we can even now start to check if these chaperones are direct interactors of tau using, for example, a proximity ligation assay that detects whether a hit gene is close to tau, or even do a specific conformation of tau. And we find both some of the chaperones, for example, interact both with wild type and mutant tau, but others seem to be selectively interacting in mutant neurons with tau. And we can even ask, where is it interacting, and validate these things in patient brains. So with that, coming back to our initial questions, we can use deep mutational scanning to ask which protein states are disease relevant. Hopefully soon in cell types such as neurons, we can understand what controls formation of these using CRISPR-IA modifier screens. And then, of course, the really important next question is to ask, how do these protein states cause disease? How do they affect cellular processes that then lead to disease? And again, a combination of deep mutational scanning and CRISPR modifier screens will hopefully tell us that. So with that, I'd love to thank my lab, who have been a wonderful collegial cooperative group of people during this difficult pandemic time, our collaborators and funding sources. And I'm looking forward to discussing more with you and taking your questions. Thank you.